Hello students, this is Dr. Ravi Seni, and today we are going to discuss about the resting membrane potential. And that is really important topic because most of the mechanisms which are really basic in nature to understand the normal human physiology, there are some basic principles, and it is one of them resting membrane potential. Now come to the various factors which affect net rate of diffusion of ions through the membrane. So what are the various factors which affect net rate of diffusion of ions? To start resting membrane potential, first we should know about some other concepts which are related to the generation of or genesis of resting membrane potential. First, we should know what are the various factors which affects diffusion of ions through the membrane, in which the first one is the concentration difference. In earlier classes or in lower classes, we have studied that concentration gradient. Basically, if across the membrane, this one is the membrane, and across the membrane, if there are concentration difference, if some, if at some, if this side there is a high concentration, on this side there is a low concentration. So this concentration difference creates a force. We call it chemical gradient, or the which is produced. This force is basically produced due to concentration difference, and this gradient is responsible for the movement of the ions. So in case of factors which affects the rate of diffusion of ions, the first factor is the concentration difference. So there should be a concentration difference through which a gradient will be generated, a force will be generated that will be responsible for the movement of ions. Similarly, the second factor is the electrical potential difference. If across the membrane there is potential difference, potential difference, like outside if there is a positive and inside the membrane there is negative, so definitely there is a difference in potential, there is difference in voltage or ions. So this potential difference also create a gradient or a force which is responsible for the movement of ions. Now we have got two forces which are responsible for the movement of ions. In this image, this one is a typical ion channel. Basically, it is a transmembrane protein. Ion channels basically are formed by the transmembrane proteins. And the ion channels basically have two important properties. They have selective permeability, like some channels have selective permeability, means they will allow specific ions only. So ion channels should have selective permeability. Now come to the two forces. The first one was the electrical gradient, and the second one is the chemical gradient. So these two forces are responsible for the movement of ions or the diffusion of ions through the membrane. It might be possible that both the gradients could be in the same direction, or could be in opposite direction. Obviously, if these two gradient or these two forces are in same direction, then net movement will be in their direction. But in case of they are in opposite directions, then then the net movement of the ions would be in the direction of the greater gradient. If the chemical gradient is greater, then the ion will move in the chemical gradient direction, and if the electrical gradient is more than the chemical gradient, then the net movement of the ion will be in the similar direction of the electrical gradient. So basically, we understood about the two forces which causes net movement of the ions. In this slide, we have got the effect of concentration. Effect of concentration, a difference which affects diffusion of ions through the membrane. So as we know that the net diffusion into the cell is proportional to the concentration outside minus the concentration on the inside. So the net diffusion will be in the it will be proportional to the concentration difference. And CO is the concentration outside, and Ci is the concentration which is present inside the membrane. So the net diffusion will be proportional to the concentration difference. In case of electrical potential difference, we have a specific equation to calculate the potential difference. If a body is having 37 degrees Celsius temperature, then the electrical difference or the potential difference could be calculated for any univalent ion. Let's say example here, sodium and potassium. The potential difference for both these ions could be calculated by the Nernst equation. This is the modified Nernst equation. Here we are having C1 and C2. C1 is representing the concentration on side 1 and concentration on the side 2 of the plasma membrane or the cell membrane. Through this Nernst equation, we can calculate the electromotive force or potential difference for any ion. Now come to the another equation, because in case of cell, definitely there will be presence of multiple ions. So in a combined form, we'll have to calculate the EM, which is the total membrane potential. That could be calculated by this equation. Here we are having potassium, sodium, and chloride ions. We can calculate the total membrane potential by this Goldman equation or Goldman-Hodgkin-Katz equation. Here P is the permeability of the membrane to each ion, and C is the concentration of respective ions on inside or outside. CO is representing a concentration outside and Ci is representing concentration inside for respective ion. Like here it is for uh, th- this P is the permeability for potassium ion, and this one is the concentration inside or concentration outside for potassium. Similarly, here is the permeability for the sodium and the concentration of sodium uh, inside and outside. Similarly, this is the permeability for chloride, and this is the concentration of chloride outside and inside. So by this equation, which is called Goldman equation or the Goldman-Hodgkin-Katz equation, we can calculate the total membrane potential, because in a cell, definitely we know that there will be multiple types of ions, mainly sodium, potassium, chloride. Now come to the another equation, which is called chord conductance equation. So calculation of diffusion potential when the membrane is permeable to several different ions. This chord conduction equation could also be used for calculation of membrane potential. In the previous slide, we had Boltzmann Hodgkin Katz equation. Here we are having chord conductance equation. Like we can see that this is the Boltzmann equation or Boltzmann Hodgkin Katz equation. And here we are having chord conductance equation. Then this G is representing the conductance of the membrane to that particular ion. This G is representing conductance of the membrane to the particular ion, and E is representing the equilibrium potential of the ion. This one is the conductance, total conductance. And this one is the conductance for a specific ion. In this case, this one is for potassium, this one is for sodium, and this one is for chloride. And E is the equilibrium potential of that particular ion. In case of this one is the equilibrium potential for the potassium, for the sodium, for the chloride. And by this whole equation, which we are calling it chord conductance equation, we can calculate the membrane potential. Now come to the our main topic, which is the resting membrane potential. Till now, we have studied about the ion channels. These are the transmembrane proteins which are responsible for the movement of ions. There are sodium channels, potassium channels, calcium channels, and chloride channels. And after that, we studied about the Nernst equation, through which we calculated the equilibrium potential for particular ion. And after that, we had Boltzmann-Hodgkin-Katz equation, through which we calculate the final membrane potential. And it is also called resting membrane potential. Now come to the definition. Resting membrane potential, it is an electrical potential difference across the membrane of the all living cells, with the inside being negative in relation to the outside. So basically, this one is a potential difference which is present in all living cells, and there is a negative in relation to the outside inside the cell. This potential difference, we call it membrane, resting membrane potential, because the cell is in resting state. It is also called transmembrane potential because it is represented by the across the membrane. So we call it transmembrane potential or resting membrane potential. And this potential is produced by the movements of the ions. They arrange themselves along the outer or inner surface of the cell membrane. There will be a movement of these ions through the membrane. They will arrange themselves along the outer and inner surface of the cell membrane, and there will be a potential difference. But cell is in resting state, so we call this potential difference as the resting membrane potential. The other name is the transmembrane potential. Now come to the origin of the normal resting membrane potential. And this, the first is the sodium potassium pump. So this is a sodium potassium pump which is responsible for the movement of 3 sodium ions outside, and there will be a net movement, or sorry, there will be a movement of 2 potassium ions inside the membrane. So basically sodium potassium is transferring 3 sodium ions outside the cell and 2 potassium inside the cell, and it requires ATP. There will be a consumption of energy, or energy will be required for the movement of these sodium and potassium channels through the sodium potassium ATPs. So finally, more sodium ions are being pumped to outside than the potassium to the inside, and which finally causes or creates negativity inside the nerve membrane. Here we are taking an example of nerve membrane. It could be cell membrane. So finally, the net movement of these ions, 3 sodium is transferred outside and 2 potassium are entering. There will be a generation of potential difference. It could be around -4 millivolt. This amount of potential difference is being generated by the sodium potassium ATPs, because due to movement of 3 sodium ions, there will be positive charge outside the membrane, and inside there will be a less positive, or we call it negative. Here we are having less positive inside the cell, more positive outside the cell. So less positive could be called negative in relation to this. There will be a positive outside finally, and inside the cell there will be negative potential. Now come to the other factors. The first factor was the sodium potassium ATPs in genesis of or generation of resting membrane potential. The other factors are the potassium diffusion potential and the sodium diffusion potential. In sodium potassium pump, or through the sodium potassium pump, both the ions were moving. 3 sodiums were moving outside of the cell, and 2 potassium were moving inside the cell. But there are sodium channels or potassium channels are present in plasma membrane or cell membrane. So due to movement of the ions through these respective channels could also generate diffusion potential. In case of potassium, we are calling it potassium diffusion potential because potassium is continuously diffusing through the potassium channels. Similarly, sodium is also diffusing through the sodium channels and is responsible for the production of sodium diffusion potential. But one important point which should be noted here that at resting state, mainly sodium channels are closed. So finally, there will be a net movement of potassium outside the cell. Potassium will move outside the cell and will generate potassium diffusion potential. Some amount of sodium could be diffused inside the cell and it will generate sodium diffusion potential. If we'll calculate the diffusion potential in case of potassium, it could be around minus 94 millivolt. In case of sodium, the potassium, the diffusion potential would be plus 61 millivolt, which is calculated by the Nernst equation. Nernst equation. Through the Nernst equation, we calculated the potassium diffusion potential. It is around minus 94 millivolt, and the sodium diffusion potential it is around plus 61 millivolt. So by using Nernst equation, here we put the concentration of sodium outside or inside, and finally calculate the value. It is around plus 71 millivolt. Actually, in this slide, we are using the formula Nernst equation, and here we are calculating the value for cardiac muscles. And in the previous slide, we calculated these values for nerve cells. Formula is same. Similarly, we calculated the potassium diffusion potential through the Nernst equation. It could be around minus 94 millivolt. Now come to the other factors. The contribution of potassium diffusion potential and the contribution of sodium diffusion potential. In previous slide, we studied about the sodium potassium ATPs. through this sodium potassium ATPase or sodium potassium channel, there were both the ions were moving. Sodium were moving, 3 sodiums were moving outside, and 2 potassiums were moving inside. But there will be a movement of sodium ions through sodium channels, and potassium ions will also move through potassium channels. Apart from the sodium potassium ATPase, these ions will also move through these respective channels, and they will generate potassium diffusion potential or the sodium diffusion potential. In this image, it could be around, we can see that the diffusion potential for potassium, it is around minus 94 millivolt. That is calculated by the Nernst equation. And in this case, basically here we are having an example of nerve fiber. So in nerve fiber, the sodium diffusion potential, it could be around plus 61 millivolt, and that is also calculated by the Nernst equation. So apart from sodium potassium ATPase, these two factors also contribute to the resting membrane potential. This is the Nernst equation. After putting the concentration of sodium outside and inside, we calculated the membrane potential or the diffusion potential. For sodium, it is around plus 71 millivolt. In case of cardiac muscles, in case of nerve muscles, it was around plus 61 millivolt. In nerve fiber, this one is calculated in cardiac muscles. Similarly, we calculated the membrane potential for potassium or the diffusion potential for potassium to Nernst equation, and the value, it is similar, minus 94 millivolt in case of cardiac muscles and in nerve muscles as well. So finally, we have got the values for both potassium and sodium, the diffusion potential for potassium and sodium. Now we'll put these two values in chord conductance equation. In this image, we can see that finally there is a tug of war. Finally, there is a tug of war between the sodium and potassium. Basically, both ions try to make the resting membrane potential similar to their respective values. So this one is the diffusion potential for sodium. This one is the diffusion potential for potassium. So both are trying to make a resting membrane potential towards each other. Now, who will win the war? We calculate through this chord conductance equation. This is the chord conductance equation. To Nernst equation, we calculated the value of this potassium diffusion potential. Here we calculate the sodium diffusion potential. These one are the conductance values. And through chord conductance equation, in case of cardiac muscles, the resting membrane potential is calculated around minus 90 millivolt. In this slide, we are using chord conductance equation for calculating the resting membrane potential. But in previous slides, we have also mentioned that Boltzmann-Hodgkin-Katz equation could also be used for calculating the resting membrane potential. The respective diffusion potentials or the membrane potential for sodium and potassium is calculated through the Nernst equation. And in this image, we calculated the resting membrane potential for cardiac muscle, and it is around the minus 90 millivolt. In case of nerve cells, this value could be around minus 70 millivolt. In skeletal muscles, similarly, it is minus 90 millivolt. In cardiac muscles, it is minus 90 millivolt. So finally, we have got the resting membrane potential, in which sodium potassium ATPs, sodium potassium channel, and finally diffusion of potassium, diffusion of sodium, and sodium potassium ATPase. These three factors are mainly responsible for generation of resting membrane potential. But a point should be noted that at resting state, most of the sodium channels are closed. So there will be a main contribution by the potassium diffusion, because these potassium channels are having leaky property, or they are also called leaky channels. Sometimes they are also represented at the leaky sodium potassium channels. So finally, these are the factors which causes generation of resting membrane potential through various equations. The commonly are the Nernst equations. The second one is the cord conductance equation, and the other equation was the Boltzmann-Hodgkin-Katz equation.